Hello and welcome back to another year of The Researcher Behind the Image, where I, Jessica Anania, speak to the researchers that submitted into our calendar competition for 2025. I hope you've all had an opportunity to pick up a calendar or download a copy from our website where it's also available. Today I am really glad to be joined by our winner, Eva, who will be taking me through her research. Welcome, Eva. Would you like to introduce yourself a little bit?
Hi, everyone. Thank you. I am a PhD student at the University of Oslo. More specifically, I am working at the Greif lab, and my main focus is antibody-antigen interactions and understanding their behavior from the structural perspective.
Brilliant. And I'm personally excited to learn a lot more about the structural side of things and the computational side of the work that you're doing. So we have a wonderful video here provided by Eva that will introduce her work, and then we will learn a lot more about the specifics.
Amazing. So I hope this video can introduce you a bit to the structural world. And this is the full antibody structure. You might know that it's a big molecule. It consists of heavy and light chain. And on this video, you can see that green is heavy chain and pink is light chain. But when you work in computational biology, you need smaller system. Usually you focus only at the ending of the arms and specifically at the CDRH3 regions. CDRH3 regions are especially important for binding. That's why we are so interested in them. And binding is one of the main qualities of antibodies that made them such great drugs. They have a specific and precise binding to antigens. And this binding is what all the companies want. The binding happens in a very specific place, yeah. And the binding is defined by a loop. Loop is a very flexible region. That's why it's so hard to understand. Also, CDRH3 are very flexible in terms of sequence. So sequence are very variable, and also the structure is very flexible. And CDRH3 is the most interesting out of all the loops because it's the source of highest variability and flexibility. So if you want to understand antibody-antigen binding, you should focus on CDRH3. However, up to date, the structural rules behind CDRH3 binding to antigens are unclear, partially because it's flexibility and partially because it's variability. So my current work focuses on understanding structural features that can explain the binding. More specifically, we look into the complementary experimental and computational techniques that can help us to provide this understanding. And understanding means the features from the structural world, like interatomic interaction, surface, and general structure that can be quantitatively described and extrapolated into high throughput manner. Why it's important? Because this way we might be able to accelerate computational antibody design. And computational antibody design is important to accelerate, in general, biologic drug discovery. Being able to screen quicker antibodies prior to experiments can allow us to boost the success rate at the later stages. And this is the long-term goal of my project and my PhD research in general.
Okay, thank you for that lovely introduction to your work. How much of your work is based on computational, and do you do any wet lab as part of that? Do you actually test the computational output that you're getting to engineer the antibodies and see how closely they align?
My work is completely computational, but our collaborators, both within the lab and outside the lab, help us with experiments. For example, we were able to get SPR, surface plasmon resonance, to really evaluate the binding affinities. It was made in our lab, but with collaborators, we were able to get cryo-EM of the variants to understand better their structure and how close it is to the modeled ones. Also, our collaborators provided us with HDX-MS to get the peptide-wise resolution of the binding interface. So I would say we try to combine both, but personally, I work on the computational side.
That must be so satisfying to see how closely it matches up. So can you tell us what the actual structure is that we're looking at in our January image? Because it looks wonderful and space age, but where's the part that you're actually interested in and what's the structure? I'm guessing it's the CH3, but
Yeah, sure. So, as I mentioned, we are combining computational experimental approaches, and I try to reflect this setup here. The structure is a complex between trastuzumab, it's a blockbuster antibody, and HER2, its target. The
okay.
antibody is the pink and the yellow, let's say. Pink
Yes.
is a heavy chain, yellow is a light chain, and it's only the variable region, while the purple one is HER2, the antigen.
Yeah.
So the outer circle, it's, let's say, artistic representation of SPR data that we very
Yes.
blurry see that the binding happens. Then the circle inside is HDX-MS that shows peptide-wise resolution of the binding. Then
Okay.
coming closer, it's something that we already can model. For example, with molecular docking, that we can see already residues and more closer binding site. And the most inside circle is molecular dynamics, where we can already look into the interatomic interactions between atoms, residues within the time. So you can see like every atom and the residue represented as stick there.
No, it's beautiful and it's refreshing to get a different type of image as well.
Antibodies has very unique structure in terms of that they combine both ordered and disordered regions, which make them both very interesting target and very complex target to work with. Let's say,
That's very
yeah.
true. So you obviously work on the variable region, but do you have people working on the more ordered structural parts of the antibodies as well?
Not
So
in my lab, and
no.
plus I think there's, let's say, if we talk from the structural perspective, we have this IG domain, which is actually beta sheets,
Mhm.
and they're very stable. So you can you don't really have much to do with them because they're conserved and they're stable. So I think most people focused on loops because they're responsible for binding, and also you usually
Yeah.
need to mutate them to improve the stability. But I
Yeah.
know that there are some people who are trying something like protein MPNN, like redesign the sequence of an antibody to fold at the same shape, meaning that you can have the same fold of an antibody, but have very different sequences that form this fold. And this, I think, quite interesting from the academic research perspective. And I know that some people are doing this.
No, interesting. So to bring it back to your image, so you mentioned about the loops and things. Have you noticed in different antigens that they need to fold differently? Like, is there any patterns that you've observed with say HER2 versus a different cancer target? And the structural, is there any patterns that you've come up with in your research that you found interesting?
We do found them, and they will be in the soon-to-be publication.
Oh,
But overall, we
brilliant.
were looking into strong and weak affinity binders and
Yeah.
look at their structural features like flexibility of the loop, for example. And we did
Okay.
found
So
very
that would
interesting
be
things. Yes.
in the pink parts of your image then.
Yes. And
Yeah.
if you look at the blue part, at the pink part, this
Yeah.
is exactly the CDR3 loop.
Okay, so yeah, so you find most of the patterns in different binding capabilities in those regions.
Yes,
No,
both flexibility wise and the types of interaction that they form.
no,
And the most
I personally look forward
exciting,
to reading it.
yeah, and the most exciting part is that actually our computational results correlate quite well with experiments, and it's exactly what you want to see when you work in computational
Yeah.
biology.
Yeah, no, that's exactly, that must be hugely reassuring. So it's exciting to learn more. So if you say, well, you're looking at HER2, are you focusing on cancer targets then, or what type of targets does your lab work on?
I would say that we are more into fundamental research. Let's say we
Yes.
want to use HER2 and trastuzumab as a model system to be able to extrapolate the rules that we find within this model system on other targets and
Yes.
other systems in general. So I would say it's not exactly about what the disease or what the target, but to understand better how antibodies works, and specifically antibody antigen binding.
Yes, no, that's interesting. And your lab, I noticed on the website also uses AI and things like that to bring into the research. Do you utilize that at all, or can you extrapolate on that a little bit more?
Sure, yes, AI is widely used in my lab, especially in the computational part. I am more towards computational structural biologists in
Yeah.
the classical sense, but I also do use AI. And let's say some of the example is the structure prediction models. You probably
Okay.
heard about AlphaFold and
Yes, yes.
similar models that manage to boost the accuracy of protein structure prediction a lot. And I
Mhm.
am using models like AbodyBuilder, IgFold, also AlphaFold, and many others to try to predict and represent properly antibody and antibody antigen complexes.
Have you published any of the tools that you're working on yet? Or are they coming out soon for people who are interested in them?
Um, let's say the paper for this image is coming soon, but I
Oh,
have a few
brilliant.
other papers that address similar things. For example, we have a paper, I think it's published in MAPS, about progress and challenges in machine learning based antibody design. It's a nice overview of the field and how AI can be helpful in antibody drug discovery.
Oh,
Also, we have
brilliant.
a
So, yeah,
yeah.
people who want to learn more, that would be fantastic. So, are these sort of tools that anyone with a little bit of knowledge can utilize and implement in their own work? Are these publicly available yet, or is there anything similar out there that people can use?
For developability, we provide both datasets and
Mhm.
Python scripts. So if you will go to Zenoda and GitHub, you will be able to access the Python scripts and
Yes.
rerun the code and use our code for their own research.
Mhm.
And regarding tools that I use for this image, such as a bodybuilder and others, they're not from our lab, they're from other labs, and they're also open source. And you can use, for example, a bodybuilder both as a web server and standalone tool in your own computer.
No, brilliant. So anyone who's interested into getting into a little bit more of the computational side of antibody research now has some new tools potentially that they can look into. How did you get into this field? Were you always interested in the computational side? What drew you to antibodies then?
To be honest, I in high school I did literature. So
Oh,
actually, I
okay.
was interested in Russian literature. And my first conference publication was about archetypes and mythologems in Russian literature of 19th century.
That's a big jump. Big jump.
Yeah, and I moved to STEAM because in one of the summer schools, I saw this beautiful 3D visualization of molecules, and I thought that actually literature and art is very similar to science. So why to have your life easier? Just go to STEAM and combine science with art when you can. So
And
that's how
how
I moved there.
perfect that you're part of our calendar then, being that we're trying to show people how beautiful the images that come out of science are. So that a lot of our viewers are early career researchers, and we're all really encouraged to try and learn how to code and have both sides of the research wet lab and dry lab skills. What advice do you have for any of our early career researchers out there as someone who is more advanced in these techniques?
I would say that the main advice is not to be afraid to try,
Mhm.
especially computational tools, because you don't lose much. It's like there are many web servers that you can try and then analyze the results. It does not cost you anything, and you can do it in your free time. And when you are using a web server or something like Google Colab, then you don't even need much computational resources. So it's quite easy to start, and then you can familiarize yourself more and more with each step forward.
No, that's brilliant. Thank you for those encouraging words. Thanks for joining us again. I hope you've enjoyed learning a little bit more about computational biology with Eva, as I did. And I will be joined each month by our researcher behind the image, so I hope you can join me along the way.